In a groundbreaking discovery, researchers recently found microscopic, jagged shards of plastic embedded in every single examined human testicle. This finding arrives as global male sperm counts and fertility rates continue a steady, multi-decadal decline. For years, the scientific community focused on environmental factors like pesticides and heavy metals to explain this trend, but these factors failed to account for the full scale of the drop. Over the last half-century, the global environment has been saturated with billions of tons of non-degradable waste. Finding these fragments inside human reproductive organs demonstrates that environmental plastic pollution has become a direct biological crisis. At the University of New Mexico, Dr. Xiaozhang Yu hypothesized that these microscopic fragments could be the unidentified factor driving the global fertility decline. The investigation was prompted by a colleague's discovery of microplastics in human placentas, raising the question of whether male reproductive organs were equally exposed. To reach these tissues, particles must breach the blood-testis barrier. This dense layer of cells is designed to filter out toxins and protect the reproductive system. However, plastic particles measured in nanometers, a billionth of a meter, are small enough to slip through these defenses. Proving that plastic has breached this biological fortress changes our understanding of how modern synthetic materials interact with the human body. The study utilized 23 human tissue samples from autopsies and 47 canine tissue samples obtained from local veterinary clinics. Dogs serve as an effective comparative baseline because they share our immediate environment, consume the same water supply, and possess a biological process for sperm production that mirrors our own. The researchers used a specialized chemical treatment to dissolve away the natural fats and proteins of the samples. The remaining liquid was then spun in a high-speed ultracentrifuge, a machine that uses extreme rotational force to separate materials by density, forcing any solid plastic fragments into a concentrated pellet at the bottom of the tube. To identify the material, the pellet was heated to 600 degrees Celsius inside a mass spectrometer. A mass spectrometer is a tool that identifies chemicals by measuring the mass-to-charge ratio of their ions. By analyzing the gas released as the plastic burned, the team could identify its unique chemical signature. This rigorous extraction process was the only way to isolate invisible pollutants that have become deeply integrated into dense biological tissue. The analysis confirmed that microplastics were present in every single human and canine sample tested. The data shows average plastic concentrations. Dogs averaged 122 micrograms per gram, while human tissue contained nearly 3 times that, at 329 micrograms. The chart also breaks down the specific polymers, mostly polyethylene from grocery bags and bottles, followed by PVC from household plumbing. The same materials produced for industrial and consumer convenience are now bioaccumulating in the body's most sensitive organs. The presence of the plastic is established, but the broader concern is the actual physical damage occurring inside the tissue. While the human samples were chemically preserved and could not be used to measure sperm quality, the canine data showed a direct correlation. Higher concentrations of PVC in the tissue were associated with significantly lower sperm counts. This damage occurs because PVC is an endocrine disruptor, a substance that interferes with the body's natural hormonal system. In the testes, it releases chemicals that block the specific biological signals required for cellular reproduction. There is also a significant timeline problem. The human samples came from men whose average age was 35 in 2016. Their exposure began decades ago, when global plastic production was much lower than current levels. Today's younger generations are maturing in an environment where plastic saturation is exponentially higher than it was for the men in this study. While researchers advise against panic, the data suggests that a century of plastic use has progressed beyond environmental pollution to the internal modification of human biology.